Hello, my name is Mohamed Beheiry, and in this presentation I will be discussing image processing using virtual reality. I'd first like to start off by thanking Sophie Allard and the entire RTFM team for this invitation. It's indeed a great pleasure to have this venue to present this work that's been ongoing for the past few years. It is the subject of a collaboration between the Institut Pasteur, the Institut Curie, and Paris Sciences et Lettres. To begin this presentation and to introduce the topic, I'm going to return to the basics of image processing. The goal of image processing is always to extract meaningful information. Of course, the methods that we can use to extract this information vary in complexity, and the images that we analyze also vary in complexity. But we always seek to extract information such as distances, object detections, correlations, events, etc. In this slide, I present an example of the information that we may be seeking to acquire from an image that we're analyzing. On the left is an acquisition from a multifocus microscope of 20 nanometer tracer beads that have been microinjected in a eukaryote cell nucleus. The multifocal microscope is a very brilliant device actually. It allows you to image 9 individual focal planes simultaneously and to have a readout on your camera which is in the form of a 3x3 image matrix. What we seek to understand in this acquisition is the heterogeneous dynamic behavior, the spatial dependent dynamic behavior of diffusing particles in the cell nucleus. The first step in analyzing this type of data is to first constitute 3D trajectories of these 3D tracer beads through single particle tracking techniques, and that's what you see in this middle panel here. Next, what we do is we extract a map of the dynamic parameters that dictate the movement of these trajectories, such as the diffusion, for example, or the drift. And what we see on the rightmost panel is an octree, or this Minecraft type representation of the diffusion coefficient, the spatially dependent diffusion coefficient inside the eukaryote cell nucleus. While this representation does give us a feeling of the dynamics exhibited by these tracer beads, such as the multi-scale diffusive behavior, which is very characteristic of the dynamics of the cell nucleus and its very multi-scale physical architecture, this representation is lacking. It is lacking notably due to the difficulty in perceiving this volumetric information in a very intuitive fashion, as it's very difficult to really get a feel for what's actually going inside the cell nucleus when we visualize like this. This difficulty in interpretation can be traced to a simple realization. We are visualizing inherently 3D information on a 2D screen. In fact, when we look programmatically at what's going on, the computer knows that all this information is 3D. We just communicate it in 2D by a series of matrix projections. A perceptory limitation arises in this conversion of 3D to 2D information. It can also be described as an interpretation gap. A potential remedy to this gap is stereoscopy. The use of stereoscopy dates back a very long time. It is nothing new. It relies on displaying 2 images of the same scene to each eye that have a slightly different positional and rotational offset. In essence, with stereoscopy, we're mimicking the way in which our actual eyes perceive 3D objects in the real world. It permits immediate and natural perception of 3D information. There have been many attempts of commercializing stereoscopy in the past, all with limited traction. Today, however, we have reached a certain level of technological maturity that makes useful stereoscopy available for the masses. The latest iteration of virtual reality is one of the embodiments of this technological maturity. It involves wearing a specialized headset that makes full use of stereoscopy, but also combines precise positional tracking of the headset and handheld virtual reality controllers that can be thought of as 3D mice, essentially. Wearing the headset, the user is totally immersed and transported into an entirely artificial computer-generated environment that can be interacted with in 3D. Numerous commercial VR headsets are currently available at prices that render them easily accessible for scientific laboratories. A question that you may be asking at this point is whether your research can benefit from this technology. To answer it simply, virtual reality technology can be useful if you deal with 3D data. In the context of this presentation, this specifically means whether you deal with images describing 3D morphologies, whether you perform measurements in 3D, or whether 3D dynamics are being investigated. In the remainder of this presentation, I will present two projects that we have developed that allow you to take full advantage of virtual reality in analyzing two distinct data types. The first data type that we will look at are point clouds. On the slide, you see the main contributors to this project in addition to myself. Localization microscopy is synonymous with point clouds. It involves, as many of you know, detecting individual emissions of fluorescing molecules, in so doing allowing us to beat the optical diffraction limit and observe nanoscopic structures. In this slide, we see how we presented data from a localization experiment dating from 6 years ago, specifically data from a combination storm and palm experiment where the yeast cell wall is tagged, as well as a tubulin filament that passes between them. When we observe this same data in a virtual reality context, we get an entirely different perspective. Basically, as you see, with virtual reality, we can enter inside the data, literally, and observe the 5-micron size yeast from the inside out and perceive data that may not otherwise be obvious. But let's think about the notion of point clouds a bit differently. We see that point clouds can have spatial coordinates in the x, y, and z dimensions, but they can also have additional dimensions that can be directly measured in experiments or through post-analyses. These include localization precision, timestamps, trajectories, intensity values, and even molecular orientations. How can we faithfully view all this information at once or in a logical way? Techniques such as PCA, principal component analysis, and embedding are very useful in this regard, although in using them we lose fidelity to ground truth as projected dimensions are arbitrary. There is a need, therefore, for users with localization microscopy data to manually intervene in their multidimensional data and directly observe their dimensional interdependencies. The idea here is to allow users to enter their data and perform human-in-the-loop analysis, allowing them to directly be involved in the analytics process instead of outsourcing it entirely to an algorithm. With this motivation, we developed an open source tool called Genuich that allows users to do exactly what I described in the previous slide. Point cloud data can be loaded, dimensions can be screened rapidly through a simple user interface, and the interdependencies between dimensions observed. All of this can take place in a desktop or virtual reality context, virtual reality giving the added benefit of a more natural spatial perception. Genuich is particularly useful in treating dynamic data, such as single particle tracking data, as we see in this film. It may also be interfaced with external Python and MATLAB scripts to perform specific forms of analyses. For the next topic of my presentation, we'll move on to image stacks. The contributors to this work are shown on this slide. When we think of 3D microscopy today, a number of imaging techniques come to mind. These include confocal microscopy, which allows us to image a vast variety of different specimens, ranging from individual cells to entire organisms. Light sheet microscopy gives us very high spatial temporal resolution, so very useful for imaging dynamic processes on an individual cell basis. Multifocus microscopy is very good for imaging dynamic processes in certain application domains, such as imaging yeast, which we saw in a previous slide. And of course, electron microscopy gives us unprecedented spatial resolution at the expense of a very high or a very low signal-to-noise ratio. So basically, the message which is communicated in the slide is that there is a vast variety of 3D imaging techniques. We have the means of generating this data that has the information that we're looking for in the context of image processing. However, the means of properly visualizing and interpreting this information is lacking, and that's what we seek to address with the tool that I'll present in the following slide. DIVA is a tool that takes as input any native TIFF image stack or hyperstack and creates a 3D reconstruction that can be visualized in a desktop mode or, more interestingly, in a virtual reality context. The 3D image stack can be easily modified in real time using a simple transfer function interface included in the software. Using the virtual reality controllers, as you see in this film, we can directly enter inside the data with our hands and select regions in 3D with individual pixel precision. And we can also add annotations if we so choose. The software is freely available on the site diva.pasteur.fr. And in the following 2 slides, I will present 2 concrete applications where we are using the tool. In collaboration with the group of Uwe Maskos at the Institut Pasteur, one of the questions that DIVA can help in answering relates to neuron tracing. Specifically, by tracing individual dendrites in virtual reality, we can gather information that allows us to quantify neurodevelopment and helps us answer questions related to neuron maturation. So why is it useful to do this in virtual reality? Just observing the data in this film, we see that it is extremely noisy. However, we know that the user understands and is trained to recognize what is a dendrite and what isn't, and allowing them to manually intervene in this exceptional 3D context allows them to distill the information, identify what is a dendrite, trace it, and effectively distill the information in this image, which they can later exploit through post-analysis techniques. A second application using the DIVA software relates to clone identification and selection in the developing larva brain. This is a collaboration with the group of Pauline's Speder at the Institut Pasteur. Pauline's group seeks to answer how glial cells in the brain regulate neural stem cell proliferation during development. One clue to understanding this is to identify the placement and morphology of glial cell clones and their relations to one another. In this movie, the different colors correspond to different clones. As you can see, clones have entirely arbitrary 3D shapes, and imaging techniques are frequently subject to noise and artifact structures. Using DIVA's virtual reality context, the user, with their expertise in the topic, can identify and literally paint over the entire clone and export this information for post-analyses. Now time to conclude this presentation with a few final words. So you may be asking, how can I access the software tools that are presented? So they're both available at the sites that are listed on this slide. And an important thing to note is that you do not require a virtual reality headset to use these tools. You can use them as a simple 3D viewer on any Windows-powered computer. So that concludes my presentation. Many people have been involved in the development of the projects that I described over the course of the presentation. The projects are notably housed in the group of Jean-Baptiste Masson at the Institut Pasteur and the group of Bassam Haj at the Institut Curie. And of course, we need to thank all the financiers that have also contributed to this collective effort.